Good morning, biology students, and welcome to class today. I'm not going to be here today, so here are your subplans, here are your directions, here is your direction to guide you through the class period today. Like we told you last time, today is a big time practice day for protein synthesis. Okay, so make sure you do your best to really try to work to understand this stuff and get some solid practice in. First block, I'll be checking in with you around 9 o'clock, I hope. All right, hopefully FaceTiming around 9 o'clock. And second block, FaceTiming probably a little sooner than that, probably mid-class, okay? But you can always email me if it needs to be a little sooner, or just FaceTime me if it's a little sooner. So we'll be chatting with you, helping you out if you have any questions. But here's what I need you to do. First of all, you need to grab one of the baggies, all right? One of the DNA puzzle piece baggies, and you're going to go ahead and you're going to practice transcription, that first major process in the whole major process of protein synthesis. So you're going to go ahead and the directions are on the subplans for 214, the sheet that your sub will have, okay? I'll also link this sheet to the Nemaha Valley Science website, which you can see we are at right now, okay? So it'll be on the Unit 6 page. Grab a bag of the DNA/RNA puzzle pieces. Build a DNA molecule that is 8 nucleotides in length. Build a DNA molecule, 8 nucleotides in length. Go ahead and model, all right, make believe, imagine that RNA polymerase is attaching to that DNA molecule, separating it, and then I want you to add your complementary RNA nucleotides. Now note, you're not going to add RNA nucleotides to each DNA template strand. You'll only use one template strand, okay? You'll only use one side of that DNA to complement the RNA nucleotides too, all right? So go ahead and build your mRNA strand, carry out transcription, okay? And then just move that mRNA sequence out. Your mRNA sequence should end up being 8 nucleotides in length as well. After you've done it once by yourself, you're going to buddy up, okay? So you'll build another DNA molecule. Your buddy will build a DNA molecule, trade, and then transcribe theirs, all right? I want you to do that, how many more times? One more time, okay? So you're going to end up doing this transcription process with the puzzle pieces 3 times total. The next thing, grab one of the directed readings from 10.1 and complete this, okay? Some basic practice, all right, vocab practice, the steps of the entire protein synthesis process on the back to this practice. So go ahead and complete this once you get done with the puzzle pieces. And then the kicker, the one that you will have questions on, I hope. Our protein synthesis practice page. Okay, we're actually going to carry out the entire process of protein synthesis. So once you're done with the directed reading, grab this bad boy and get to work on it. This sheet and this sheet will both be on the small little table in front of the smartboard, as will the, let me find it here, amino acid chart. Okay, this guy, but it'll only be in a half sheet form. All right, so you need one of those as well. This comes in handy when you are translating the mRNA sequence into the amino acid sequence. All right? So the gist of this thing right here is you're going to go ahead and you get a DNA molecule. So there's the template strand that you are going to transcribe into mRNA. So transcribe it into mRNA, all right? And then you will translate that mRNA into the amino acid sequence. All right? So for instance, TAC, translated into mRNA, that is UAG. UAG. And UAG, if we go to our chart over here, UAG, no, not UAG, sorry, AUG, my bad, AUG. AUG is methionine, and methionine is the start codon. So we would start protein building at that point. The next little guy on here is AAA, and the mRNA for that would be UUU. So 3 U's, U, 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 it'd be phenylalanine, okay, phenylalanine, all right? So you'd write that down. You can abbreviate it if you want, but I want you to write those amino acids right here on this line and this entire amino acid sequence until you get to the stop codon, all right? Remember, the mRNA molecule, it's read in sequences of 3 nucleotides. Those are called codons, and each one of those codons encodes for a specific amino acid that you'll write down this line. So you've got 7 practice problems that you complete with this. Alrighty. I think that does it for your homework and what I want you to do in class today. Not your homework, I guess, but that does it for direction for class today. Like I said, first block I'll try to check in with you around 9 o'clock or so. Second block a little bit earlier. Be diligent. Get as much practice in as you can, okay? That way when I do come back on Tuesday and we're hanging out with each other, we can make it a little further before we dive into the video walkthrough for 10.2. You will have no homework for over the weekend. Your only homework is to try to understand this material a little better, okay? To finish the practice if you need to go back and watch the video again if you need to read back through the textbook if you need to look through your notes if you need to, all right? So just a lot of studying. See you in class via FaceTime. I'm excited to test it out, all right? Talk to you later. Bye.